Hello everyone. What I want to talk about today are proteins. And specifically, I want to talk about the structure of proteins. But before we do that, let's put proteins in the context of the human body. So this pie chart here represents the composition of the human body. And about 70% of the human body is composed of water. Then we have about 5% of the body that's composed of ions like sodium and potassium and calcium, and also small molecules. And then the remaining about 25% are what we call macromolecules. These are large molecules. And when it comes to macromolecules in the human body, we have proteins, nucleic acids, which represent DNA and RNA. We have carbohydrates, and we have lipids. And out of these four macromolecules here, three of these, structurally speaking, are what we call polymers. So first of all, what is a polymer? A polymer is a molecule that's made up of a string of individual units. And this necklace up here represents a polymer. This whole thing is a polymer, and it's made up of these individual units. Now, what do we call these individual units? What do we call this individual bead right there? This individual bead right there? Well, generically, we call those monomers. Now, out of these macromolecules here, three of them are structurally polymers. Do you guys know which three are polymers? Well, we have proteins. Nucleic acids are a polymer, and also carbohydrates. Carbohydrates are polymers. And like I mentioned, we're gonna cover all of these macromolecules and the structure of them throughout the course of this class. But we're gonna focus on proteins today. So my question is, if this necklace here represents the structure of a protein is a polymer, then for proteins, what is the monomer? What are these individual units that make up a protein? And the answer to that are amino acids. Proteins are made up of a string of individual amino acids. And this is the generic structure of an individual amino acid. You have a central carbon, and coming off that carbon, you have a hydrogen here, but then you have what's called an amino group with a nitrogen in it. You have a carboxyl group. And then lastly, you have this group that's represented by the letter R, and that's called the side chain. And there's 20 different amino acids that make up the proteins in the body, and it's going to be the side chain that differs between those 20 amino acids. So the side chain is what's going to differ between the amino acids, and it's going to determine the individual chemical properties of those individual amino acids. So there are 20 amino acids that make up the composition of all of the proteins in the body. And these are all 20 of those amino acids here. And what they've done is they've organized these amino acids by their chemical properties. And specifically, it's the chemical properties of the side chains that give the amino acid its chemical property. So let's take a look at some of these, and let's start here down at the bottom. what these 5 amino acids all have in common is that their side chains are all electrically charged, and that makes them hydrophilic. So let's take a look at one of these right here. Let's take a look at aspartic acid. First of all, there's the central carbon. There's a hydrogen coming off one of them. There's the amino group. There's the carboxy group. And this right there is the side chain. And what you'll notice here on this side chain is that there's a little electrical charge associated with the oxygen. And so there's a net electrical charge associated with the side chain, and specifically in this case with aspartic acid, it's a negative charge associated with the oxygen. The same goes here for glutamic acid. The oxygen on the side chain in glutamic acid has a negative charge associated with it. Then these guys over here, these side chains are also charged, but these side chains have a positive charge associated with them. So lysine, for instance, has a positive charge associated with the ammonia group up top. Arginine also has a positive charge here. Histidine also has a positive charge. And a connection I want to make is that the fact that these amino acids, these side chains, have charges associated with them, that makes them hydrophilic. Okay, if something has an electrical charge, that means that it's going to be able to be dissolved in water. Because remember, water is polar, and the oxygen portion is negative, the hydrogen portion is partial positive, and what that means is that the water molecule can be attracted to this side chain. And for instance, so what part of a water molecule is going to be attracted to this negatively charged oxygen there? The positively charged hydrogen is going to be attracted to that. And then what about over here with the ammonium ion? The negative oxygen is going to be attracted to that positive charge there. And so water is able to surround these side chains and dissolve these side chains, these amino acids in water. So that makes them hydrophilic. What all of these amino acids up in this row have in common is that they all have polar side chains. So here with serine, again, here's the carbon, there's the amino group, there's the carboxy group, and this side chain here is polar. Now, the reason you don't see a negative or a positive charge here associated with the side chain is because this side chain is electrically neutral. It's electrically neutral, but what polar means is that one side of the side chain tends to have a positive charge and the other end tends to have a negative charge. And I'm not sure which one is which here, but I'm going to take a guess that the oxygen portion up here has a partial negative charge and the CH2 has a partial positive charge associated with it. So again, the net charge is neutral, but different parts of the molecule have different electrical charges. So these guys are all polar. And if it's polar, that means that it's hydrophilic, and that means that it's going to dissolve in water as well. Then we get up here, and what all these side chains have in common is that they're nonpolar. Not only are they electrically neutral, but there's not a positive or negative charge associated with either end of the side chain. And so because they are nonpolar, that makes them hydrophobic. Remember, water is polar, and likes dissolve in likes. And so water is going to dissolve other polar molecules or other electrically charged molecules. But when it comes to nonpolar molecules, those are hydrophobic. Those are going to be repelled from water. All right, let's look briefly at how we build a protein. A protein is made up of a string of individual amino acids. So how do we take these amino acids and add another amino acid onto it? How do we start to build these proteins? And that's what this is showing over here. This is a 2 amino acid chain right there, and what we want to do is we want to add an additional amino acid onto that. So how does that happen? Well, what happens is the carboxy group of one amino acid is going to form a bond with the amino group of another amino acid. And specifically what's going to happen is this hydroxide and this hydrogen ion are going to be removed. So we're removing a water molecule, and that makes it a dehydration reaction. And when you do that, we're forming a new bond between this carboxy and this amino group, and this bond is a covalent bond. So it's a nice strong covalent bond. And when we're linking amino acids together, the specific name of that covalent bond is a peptide bond. Now let's move on to the different structural levels of a protein, because this is super important. It's critical for a protein's function that it be in the proper level of structure. So the first level of structure is called the primary structure. And what the primary structure of a protein, or at this point we call it a polypeptide, the primary structure of a polypeptide simply refers to a linear string of individual amino acids. And so we have a string of amino acids that's just laid out linearly. There's no bends or folds in it, anything like that. It's just a string of amino acids. The next level of structure is called the secondary structure. And what happens in the secondary structure is you have this string of amino acids, and it starts to make these little, what we call alpha helices here. There's hydrogen bonds that are forming between some of these amino acids here, and it makes this little alpha helix. It can also make these little kinks here, and those are called beta pleated sheets. So that's the secondary structure. But the next level of structure is the one I want to focus on. That's the tertiary structure, or the three-dimensional structure, the 3D structure. And what happens in the 3D structure is that you have this amino acid, and what happens is the amino acid is it bends back on itself. Okay, it bends back and it forms this three-dimensional structure. And the reason that the three-dimensional structure is so important for a protein is that a protein is not functional until it gets into its three-dimensional structure. So it's not until a protein gets folded into its three-dimensional structure that it becomes functional. And the reason that it bends back on itself is because you have interactions between side chains here that are forming all these different types of bonds. And that's what basically allows the protein to fold back and form its three-dimensional structure. Then the next level of structure above that, and not all proteins go to this level, is the quaternary structure. And what happens there is when you have two or more proteins that are each in their three-dimensional structure bind together themselves. So this may be an individual protein in its 3D structure. This may be another one there. But to fully form this protein molecule, you have to have interactions that form between these two individual three-dimensional proteins. And that makes a protein in its quaternary structure. And hemoglobin is a great example of that, and we'll use that as an example later on here. Okay, so what we have here is a protein in its three-dimensional structure. And the way you can envision this is that you have all of these individual amino acids that are linked together by peptide bonds, by covalent bonds, and then there may be some alpha helices in there and some beta pleated sheets that are all going on right there that they're not specifically showing. But what's happening here is that the protein is now folding back on itself, and it's achieving its three-dimensional structure. And the reason it's folding back on itself and it's able to do this is because you get interactions between the side chains. So here we have an amino acid right here, and this is the side chain for the amino acid. Then we have another amino acid there, and that's the side chain for the amino acid. And what's happening is that there is a chemical bond forming between those two amino acids. And it's that bond between the side chain that's causing the protein to fold back on itself. So it's the interaction between the side chains that's folding the protein back and causing it to take its three-dimensional structure. So what I would like you guys to be able to do is to break down these side chain interactions that are causing a three-dimensional structure. And the first thing that I want you guys to do is to identify these individual amino acids. So what is this amino acid here? And how are you going to find that out? Well, you know the side chain, so you just need to go to the amino acid chart and find the amino acid with that side chain. Then I want you to find out what that amino acid is right there. And once we know those amino acids, and specifically we know the chemical characteristics of those amino acids, then we can determine what type of chemical bond is forming and linking these two side chains together. So why don't you take a second and identify these individual amino acids here. Okay, let's find this amino acid first. What amino acid is that? It has CH2, 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 NH3. And it also has a positive charge associated with it. And what do we have? That is lysine. There's the side chain. So we can go ahead and put a lysine right there. What about this guy over here? CH2COO with a negative charge on the oxygen. That is aspartic acid, or ASP. So now the question is, what type of chemical bond are holding these two side chains together? And if you go down here, these are the potential chemical interactions, chemical bonds that can form between these side chains. We can have hydrogen bonds. And a hydrogen bond occurs between polar side chains, and it's when a partially positive hydrogen on one molecule forms a bond with a partially negative atom on another molecule. Another type of bond we can have is an ionic bond. And this is when positively charged side chains bind to negatively charged side chains. That forms an ionic bond. Another type of bond or interaction we can have is called a disulfide bridge. And this occurs with the side chains of the amino acid cysteine. And what happens with cysteine is, and we'll take a closer look at this, is the sulfur groups interact and they form what's called a disulfide bridge. And this disulfide bridge, these are covalent bonds here. You can also have what are called van der Waals interactions. And these are temporary weak interactions between molecules, and we're not going to talk so much about those. But the last thing that can happen is you can have what's called a hydrophobic effect. And this is when you have nonpolar hydrophobic side chains. And if you have a hydrophobic side chain, what's that side chain going to want to do? It's going to want to be found towards the center of the protein, away from the surrounding water on it. And so they're going to tend to bend towards the center of the protein. Now, out of all of these interactions here, what type of interaction is occurring right there? Well, we have a positive charge that's linking together with the negative charge. So that is an ionic bond. That's an ionic bond. Next, let's move on to this interaction right there. And let's start by finding that amino acid right there. What's that amino acid? That is asparagine, ASN, and that is a polar side chain. And then what about this amino acid up there? That is serine, and that's also polar. So based on that, what type of chemical bond is forming there? These are both polar, and specifically though, we have a hydrogen interacting with an oxygen. Well, let's go down to our choices here. Well, I don't see any positively or negatively charged side chains. So so it's not going to be an ionic bond. These are both polar side chains. They're hydrophilic, so that rules out hydrophobic. I don't see neither of those are cysteine amino acids, so that rules out the disulfide bridges. But what we do have is we have a couple polar side chains interacting with each other. And off of one of those polar side chains, there's a hydrogen atom. So what's that going to be? We're going to have a hydrogen bond that's holding those together. And what's going on is that this is polar, and there's a partial positive charge associated with the hydrogen and a partial negative charge associated with the oxygen. And so that by definition is a hydrogen bond. Let's see, let's move on to that guy right there. So let's identify both of those amino acids. And it's the same amino acid for both of those. So a CH2S side chain. What we're looking at there is cysteine. So each of these is cysteine, and those were also polar. Now the key here is that we see these two sulfur molecules. And when you see those two sulfur molecules linked together, that should direct you right here to these disulfide bridges, two sulfurs. So this is a disulfide bridge, and that's a covalent bond that's linking those two together. And then the last thing we have is right there. Let's identify these two amino acids. And these two amino acids are the same. Those are each valine amino acids. And those are nonpolar, they're hydrophobic. So that's a valine. That's a valine. And what do you guys know about hydrophobic side chains? Hydrophobic side chains tend to fold towards the center of the protein. So the way you can think about this protein, if it's inside the cell, in the intercellular fluid, in the interstitial fluid, or dissolved in the plasma, this is all going to be water that's surrounding the protein. And what's going to happen with these hydrophobic side chains is they're going to try to bend and fold their way deep inside the protein away from the surrounding water. And that's what's happening here. Both of these amino acids are pointed towards the center of the protein here. And they're actually interacting with each other. And those interactions are hydrophobic interactions. So we have a hydrophobic effect. Yeah, and then just to kind of reiterate, why that's so important is, again, it's these interactions between the side chains that are causing the protein to fold back on itself and form its tertiary, its 3D structure. And the reason that's important is because proteins are not functional until they reach that tertiary, that three-dimensional state. Here's another example of a three-dimensional protein with different side chain interactions here. And I'm going to leave this one up to you guys. But again, you guys should be able to identify these individual amino acids and then tell me what type of chemical interaction is allowing those side chains to interact with each other. All right, and then I'd like to talk about hemoglobin as an example of these different levels of protein structure. This is a really good example to talk about that in. So first of all, this is a hemoglobin molecule. And we find hemoglobin inside the red blood cells of the body. And in fact, there's about 250 million hemoglobin molecules per red blood cell. So there's a whole bunch of these things within each red blood cell there. And what's the job of hemoglobin? The job of hemoglobin is to transport oxygen through the blood so it can deliver it to the cells of the body. And specifically, that oxygen gets attached to that iron molecule there. Okay, so let's look at the structure of hemoglobin here. Hemoglobin is made up of 4 globin chains. There's what we call 2 beta globin chains and 2 alpha globin chains. And let's see if I can just outline . It's a little too thick there. Okay, that's 1 beta chain. Something like that. That's another beta chain. That is an alpha chain. And that's another alpha chain right there. So you have 4 globin chains that are all linked together. And get rid of this here. What's important here to realize is that each one of these globin chains is an individual protein in its 3-dimensional structure. So this is a protein in its 3-dimensional structure. Same goes with all of these globin chains. So you can think of these as strings of amino acids. And there's some alpha helices and beta plated sheets in there that you can't see. But what's happening is this chain is folding back on itself in all these different areas. And the reason it's doing that is because you have these side chain interactions that are causing it to do so. So we have 4 globin chains, and each of these globin chains is a protein in its tertiary structure. And in fact, each of these globin chains is 146 amino acids long. So I need you to be able to recognize that each of these globin chains is a protein in its tertiary structure. But what happens with hemoglobin is we have 4 of these globin chains that are linked together. So what structural level does that bring us to if we have 4 tertiary proteins that are all linked together in a single molecule? That's the quaternary structure. And just to finish off talking about the structure here, each of these globin chains has a heme group associated with it, and that's what you're looking at here. This heme group is a carbon skeleton, and dead set in the middle of that carbon skeleton is a single iron molecule. And like I mentioned, it's that iron molecule that's going to attach an O2 molecule onto it. So each hemoglobin molecule in total can carry 4 O2 molecules. And if you have 250 million hemoglobin molecules per red blood cell, then how many oxygen molecules can be carried per 1 red blood cell? Well, you have 4 oxygen molecules per hemoglobin. You multiply those, and that means that each red blood cell is carrying about 1 billion oxygen molecules in it, which is pretty astounding. Okay, then what I have going on over here is this is an enzyme, and this is an enzyme that's breaking down a starch molecule. But what the point here is that enzymes are also protein molecules. And they may depict it like this, but this enzyme is actually in a complex 3-dimensional structure. And that's what's being represented up here. So you have, these are strings of amino acids, and you can see kind of some of these alpha helices there and beta plated sheets. But also what's going on is it's folding back on itself in all these different locations. And it's folding back because of those side chain interactions. And it's not until it gets to this 3-dimensional shape that this protein, that this enzyme becomes functional. So not surprisingly, the chemical bonds between these side chains are sensitive to certain internal body conditions. And if these conditions are out of range, then the chemical bonds between the side chains can break and the protein denatures. And what denaturing means is having a protein in its tertiary structure, its 3-dimensional structure, and if those side chains break, then it unravels to its primary structure. And we call that being denatured. The protein is denatured when it goes from its tertiary to its primary structure. And why is that a problem for the protein if it gets denatured? Well, it's no longer functional. Enzymes, antibodies, hemoglobin, skeletal muscle proteins, if those denature, then they're no longer able to perform their jobs. So what are these parameters, these internal parameters that we have to maintain homeostasis over to maintain protein structure? And what homeostasis is referring to is just maintaining a variable within tight limits. So what are some of these internal body conditions we have to maintain homeostasis over to maintain this tertiary structure? Well, one of those is body temperature. Okay, we want to maintain our body temperature at about 98.6 degrees Fahrenheit, which is about 37 degrees Celsius. And we have a little leeway there, but not a whole lot. And if this body temperature gets out of range, specifically if it gets too high, if that gets up to like 39.4 degrees Celsius. Then the problem with the body getting that high is it breaks those bonds between the side chains, and it causes it to unravel and those proteins to denature. And that's why having a really high fever is so dangerous, because these proteins can start to unravel, denature, and then the body just starts shutting down. What is the other main internal variable we want to maintain homeostasis over? That's pH. Our normal blood pH is about 7.4, and we have a little leeway, but again, not that much. And if, for example, the blood becomes acidic, okay, it starts getting below about 7.35, then these side chain interactions start to break down, proteins start to denature, and again, the body starts to shut down, which is obviously not good. So body temperature and pH, we have to maintain within strict homeostatic ranges to keep the tertiary structure of these proteins.